Okay, but today I'm gonna hopefully try to introduce some concept of statistics to you, which hopefully will be relevant to some of the stuff towards the end of the practical, and more generally will be useful to you as a scientist. So Massimo is going to do this in a little bit more detail later on. This is not really very important. It's in German, it's something else. So just to kind of give you an idea why this is important. So this is an example from a paper that I read a couple of weeks ago that I'm using in a tutorial for some of the third years. So this is a picture of a zooplankton tow. So they're basically dragging a net through the ocean, trying to look at some plankton. And these white things, you can see in the background, those are marine plankton. But then you've also got this blue thing here, okay? And that's a piece of synthetic fibre. Okay, it's from your clothes, you wash your clothes, fibres get washed into the ocean. As these are a problem, okay, so there are some more. So here are some more fibres. The red one, there's a blue one up here, and also these kind of white dots here. These are plastic microbeads that you put in. Well, you don't put in, but are probably in the things that you wash your face with or brush your teeth with. So they're in quite a lot of kind of cleaning products, cosmetics, that kind of stuff. And the problem is that when these get into the marine environment, they're not so good for the things that live in the marine environment. So this is the paper that I was interested in. So what they've got here is a bunch of larvae of, I think, the common European perch or something. It might not be the common. It is the European perch. And what happens is that if you put microplastics in the environment, bad things happen. So they're more susceptible to predation. Okay, so this is a control experiment here, and they've basically put the fish larvae and a predator fish in the same tank, and over time the predator fish eats the larvae. But if you put lots of plankton, lots of these microbeads in the environment, they get eaten a lot quicker. And the reason for that is kind of not entirely clear, but there are a whole bunch of things that go on. Okay, so for instance, so these are basically the control is where they've got an experiment where they've not added any microplastics, and then they've added 10,000 and 80,000 particles per meter cubed of microplastics, and then they've observed the kind of the behaviour of the larvae. So basically, all of these are measurements of how much the larvae move around, and you can see if you don't put anything in the water, they basically cross a number of lines in the box as a measure of how much they're moving around. They move a further distance, and they spend less time just kind of sitting around going, what are we doing? Okay? But I'm showing you this graph because it's important for us to basically, what they've done is they've measured, instead of just measuring one fish and counted how far it's moved, they've measured a lot of fish in each of these categories, in the control and the ones where they've added the particles. Okay? And for each of those groups, the key question is the average, when you add the particles, different to the average when you don't add the particles? They're basically two measurements. Are they the same or are they different? Okay? So this is one of the most fundamental kind of things that scientists do. Okay? We measure things and say, is this number the same as this number? Okay? And to do that, you have to have some measure of the average value and also some measure of the uncertainty in that average value. Okay? Because if your measurement of the average is really like quite scattered, quite a broad range, it's very hard to say if it's different or the same from something else. Okay, so this is why we do things like working out the standard deviation, which you might have heard of before, and the mean. So I'll just also to point out where you can go wrong doing this. So this is a statement of the paper, and this is why this paper was so important. The reason it's important was because instead of just dumping loads and loads and loads of microplastics in, which would obviously kind of overload the fish with stuff, because they basically think it's food and they eat it, and then it turns out to not be very nutritious because it's kind of plastic. They went out into the environment and they measured the concentrations of plastics in the environment and did their experiments at those concentrations. So they made it relevant. Okay, so these are the statements that they made here. Okay, so they went to some Swedish coastline, because they're Swedish, not because they wanted a holiday, and they saw that the range of concentrations was from kind of 100 to kind of like 2,000, all the way up to around the kind of 100,000 kind of range. Okay, and they said the average value was around 7,000 to 10,000. Okay? So the decisions they made their control group 0, clean water, so that's looking at to basically to baseline their experiments to, so they can detect if there's any change from no pollution. Then they used the average, and they picked up the highest end of the average, which is a bit sneaky, but they said 10,000, it's a round number, that's fine. And then they went up to 80,000 for the extreme case. Okay? So the idea is that by using this average value, okay, that means that their experiments are relevant to the real world, rather than just being just dumping loads and loads of plastics in so they can see a result. So the problem with this is some of the data that they used to determine that average. Okay, so these are a whole different places which are completely unpronounceable in Sweden. Well, they're pronounceable in Swedish, but I can't pronounce them. And these are different kinds of fibres, so red fibres, blue fibres. The milky white spheres are the microplastics, and then some black or transparent ones. So this is the total sum. That little Greek sigma means the total of particles per metre cubed. Okay, and you can see that most of the numbers are around 1,000, except one number which is 100,000. Okay, so when you average all these numbers up by adding them all up and then dividing by the total number of samples, you do get an average that is between 7,000 and 10,000. So that's going to be pretty good. But is that a good measure of the most likely concentration you will find in the environment? Okay, it's not, because the mean is skewed massively by this number here. Okay, so a better measurement would be the median, which is basically you rank all of your values from biggest to smallest, then count in from the end, and the middle value is the most likely value to have. So the reason I'm showing this is because this basically means that study that they've done, they've put in their kind of average scenario concentrations that are a factor of 10 too concentrated than you would normally expect in the environment. So that's a bit sneaky of them, but it got past review and into science. So it's really important when you're dealing with data to plot it up and have a look at it. So this is that same data. So along the bottom, we've got basically the sample number, so the first sample. So quite often we would refer this to n equals 1. So the first sample is n equals 1, n equals 2, n equals 3, 4, 5. Okay, so this is just the number of samples they take. And then you can see the 11th sample is way up here. Okay, the average of all of these plots somewhere along here. Okay, nowhere near any of the data. Okay, so in this case we can see that because by plotting up the data, it's really easy to see that outlying data point. Okay, so this is a case where actually the mean, so adding all up the numbers, dividing by the numbers you have, that's not a very good measure of the most likely value. Okay, this is just another way of looking at that data. So this is plotted up as a histogram, which you can also just do in Excel. And you can clearly see that the most likely value is kind of way over here, around 1,000. Okay, the value that they used was this value. Okay, that's clearly wrong. Okay, so this is why it's important to have a good understanding of the statistics that are used, even at the basic level of working out the average value from a set of measurements. Okay. Okay, so these are these common values that we're using here. So just go through and explain a little bit of some of this terminology for you. So the mean is quite often the mean of the variable x. So if you've got, so in the example we've just given, x might refer to the concentration of microplastics. Okay, or if you're measuring all of your heights, x would be your height. Okay. This little bar above the x, that means we're looking at the mean. Okay, and the mean is defined by this equation here, and what this Greek letter sigma, kind of backwards giant E, means, it means basically to add up all of the values of x, and this subscript here, I, refers to the nth number in the sequence. So previously we had maybe 13 samples, so that would be, this I would be 1, then it would be 2, then it would be 3, then it would be 4, all the way up to 13. Okay? So from the first sample all the way up to the maximum number of samples, n. Okay, so you add up all of the numbers, and then you divide by the number of numbers that you have. Okay, so that's what that symbol means. So there's this other statistic. Okay, this is basically called the variance. So what that's doing is it's looking at how far each number is away from the mean. Okay, and I'll just draw that. So if we've got, if this is our number of measurements, okay, going up here, this is x, it's the parameter we're measuring. We might measure a first value, might be here, then we might measure something else. Okay, so we might measure a bunch of points. Okay, in this case I've measured 1, 2, 3, 4, 5, 6, 7, 8, 9 measurements. And the mean will be somewhere, somewhere along there. Okay? And what this equation is doing is it's basically looking at the value of the mean minus the value of each measurement. So it's looking at basically this distance okay, how far each of those is away from the mean value. Okay? Now, if you just measured that distance, measured all of these, and then added them all up, okay, because some of them are positive differences. If you're basically taking this number minus that number, that's a positive, but if you take this number minus that number, that will give you a negative answer. Okay? And if you've calculated your mean correctly, if you just add all those up, okay, you will always get 0. Okay? So what we do, instead of looking at the just the average of the differences, when we work out this value here, so this would be a positive number, that would be a negative number, before we add them together, we square them. We times them by their self. And that's because if you times a positive number by a positive number, you'll get a positive number. But if you times a positive number by a negative number, you'll get a positive number. So this effectively makes all of these differences positive. Okay? But they'll now be wrong because they'll be squared. Okay? So we have this term variance, sometimes called lowercase Greek sigma, just kind of like an O with a little hat on it, a jaunty hat. So that's sigma squared because this is squared. So to get back to that average distance away, okay, we are taking the square root of this number. Okay? And that will be our standard deviation. So the standard deviation is effectively the mean difference any one sample will be away from the mean of the whole population. So it gives you a measurement of how scattered your data are. So if you made this set of measurements and then you made another colour, no, I don't have another colour. Ooh, colours. If I made another set of measurements up here, you can see that the differences between the mean and these data are a lot bigger, so that would have a larger standard deviation. Okay? So if I was then plotting up a graph that had the white data, I would mark the average of the white data, and then I would put on an error bar that is that distance there is the standard deviation, so the mean difference away from the mean. Okay? Whereas if the green data, that might have a similar, it might have a bigger standard deviation error bar on it. Okay? So determining the size of these bars are important. Okay? Because, I mean, if I told you, if I asked you, is 9 bigger than 7? Is it bigger than 7? Yeah, the number 9 is bigger than 7. But if I've measured, so I've measured something about you. I don't know. I can't think of anything that's not inappropriate. Okay, the number of beers that you all had, right? So if I like split the class down the middle into kind of like male and female, okay, so I could have like beers female versus beers male, okay? And let's say the women had 9 beers, okay, on average, okay, and that's like 9. And the guys, you had 7 beers because you'd pre-gamed, okay? So, but that's not each one of you having kind of exactly that number of beers. Some of you might have had more, some of you might have had less, okay? So each of these will have an uncertainty on them, okay? Based on this difference from the mean, okay? So in this case, I can't say that this number is definitely different from this number, okay? Because the error bars overlap. So I'm not sure that this is a different number from that number, okay? So that's the idea of error bars. Okay, so we're going to talk a little bit now about other estimations of this uncertainty. So basically, if I wanted to know, if I wanted to do this experiment for, I know, the whole university, find out, you know, how much of an alcoholic each sex is, then I could go and ask everyone in the whole university. Okay, that would be tedious, right? Or I could go and take a small sample of students, okay, and just ask them. And as long as I chose those students randomly and didn't say just pick, I don't know, just pick all of the geologists, because that would be skew the results, because geologists might systematically drink more or less than the rest of the students. I can measure just the sample, mean and variance, and from that I can infer what the whole population does. Okay? So some more terminology here. So the population size, so that's the number of people in the whole university or the whole world, or whatever it is, the thing that you're trying to measure. That's big N. The sample size, so the number of people you actually ask or measure, or the number of measurements you make in the lab, that's lowercase n. And similarly, the mean and the variance are given different little symbols. That should be squared, and that should be squared as well.
What should be squared?
The population variance. Variance is sigma squared. Standard deviation is just sigma. Okay. So these are those. Yeah. So it should be like this. You see, population variance sigma squared. So they are the same equations. Yeah. So the population mean. So if I asked everybody, okay, what's their whatever it is that I'm measuring, shoe size, height, alcohol intake, and divided by the total number of people in the whole population. Similarly, if I then looked at the difference away from that mean, okay, squared it, and then divided by the total number, I would get the average variance. But I'm not going to do that because that would take me forever, so I'm just going to take a smaller sample of the population. Okay, mean is exactly the same. Now, the variance, there's this extra word in here, biased sample variance. Okay, so if you calculate the variance this way, okay, looking just at the difference between the mean and each measurement, squaring it, divided by the total number of measurements, you will get a sample variance, but it won't be directly related to the population variance. There'll be this slight bias. I'll explain that in a bit now. Okay, so this is basically some random data that I generated in Excel. So I basically made 10,000 data points that have a mean of 25, okay, and there's a variance of 100. So most of the time you'll measure quite near to the mean, but sometimes you'll measure quite far away, okay? So lots of that, it's a very large population. So if I'd forgotten what the mean and the variance of that population was, right, and I wanted to measure it by selecting some of the data points and then working out the average or the mean and the standard deviation or variance of that smaller sample. Okay, so this is what would happen. So there's that same population at the top, and this is what happens. Just look at this graph over here on the left. So as I increase my sample size, so if I just pick, say, 1 or 2 points, okay, if I take 3 points, 3 random points, okay, from that data set up there, and work out the average from that, okay, that's going to be kind of close to the average of all of those data, okay, but it's not going to be very accurate, okay, because I might have randomly selected like some points down the bottom or point at the top, okay. So in this case, when I selected 3 points, I got 80% of the way to the true mean, okay. As I increase the number of points that I pick out of that population at the top, as my sample size gets bigger, I get more and more accurate at measuring the mean. Okay, so this is kind of if you asked 10 people how much alcohol they drink, then you're not going to get a very representative result. If you asked 100 people, you're going to get much more better measure of the average of the whole population as a whole. Okay? So basically increasing your sample size gets you a more and more accurate measurement of the mean. Okay? So a similar thing happens with variance. So this is just a random sampling again. So I started out getting 3 points, then 4 points, then 5 points. And I measured the variance of those 3, 4, 5, 6, 7, all the way up to, I think, 30 points. And we kind of start to approach the correct value, okay? But we seem to be a little bit low down, okay? And if you do this again, okay, so this is basically starting with another set of random points, then starting with like 3, 4, 5, and then getting more and more points. You see, we might start high and then start to gradually get down to the true value of the mean. But the variance is still kind of not a very good measure, and it always seems to be low. Okay, sometimes it might be high because you might accidentally randomly sample from near the middle of the population. Okay, there's another example. Okay, as you increase the sample number, you get a much better estimate of the mean, but your variance is not such a good and typically underestimates. So this is basically what happens. So if I basically repeated that process of sampling and looking for small numbers of samples to large numbers of samples, then worked out how far on average I was away from the true value of the variance and the true value of the mean. And you can see that for small sample sizes, there's a systematic underestimation of the variance. Okay? And if you think about it, this is because you see all of those points at the top there, they're more clustered towards the middle value, okay? So the larger population size you're going to get, the more likely you are then, the more larger sample size, the more likely you are to sample those outlying data points. If you have small sample sizes, you're likely to miss the true spread of the data, okay? So there's this systematic underestimation of the sample variance, okay? And this is what's being shown here. So we've got the mean this way, so with small sample sizes, so these are the red points, okay, we very rarely get near the mean, which is this value here, okay, but with bigger and bigger sample sizes, we get closer and closer to the right value, okay, and we always tend to underestimate the variance. Okay, there are much more red points in this cloud under here than there are up here, okay. So what we can do is we can look at this. This seems to be quite systematic. Okay, so we want to actually take our measurement of variance and make it systematically bigger to account for this bias. Okay, and the way we do that is with this equation here. And it's actually really trivial. Okay, so if you look at the difference, so for n equals 2, so if you just have 2 samples, our variance is on average half of what it should be. If you look at 3, it's 2/3. 4, it's kind of a quarter. So that gives us a formula, okay, so that the sample variance, okay, is basically n minus 1 over n of the true population variance. So it means we can, if we know this, we can then rearrange this equation to get this, okay? Which all we're doing is this maths here. So basically our sample variance, big S, is equal to n minus 1 over n of the population variance. So when n is a big number, okay, so if n was 1,000, 1,000 minus 1 divided by 1,000, 999 divided by 1,000. That's very close to 1. So that means that if n is really, really big, big S squared and sigma squared, those numbers are very similar. Okay, so we don't need to worry about this bias too much. But if our if our number of samples is quite small, then there'll be a big difference between big S and sigma squared. Okay, so just to find out what sigma squared actually is, that's what we want to know. That's the population variance of the whole population. We basically can rearrange that top equation up there. So we divide by n plus 1 both sides and times by n. That gives us this equation. Okay, we know what this is from before. So this is the sample variance, so the difference between the mean and the measurement. Okay, average squared and then divided by the number of measurements you make. So we just combine this formula and that formula to give us this formula. Okay, because n divided by n minus 1 is the same as n over n minus 1. Okay, so this gives us basically an unbiased estimate of the sample of the population from a sample measurement. So we don't have to measure everybody, we can just measure a smaller number of people and get an estimate of the variance of the whole population. Okay, so this is kind of graphically how that cloud of data would look. So if the data is truly random, you're more likely to get a data point around the mean value than you are out towards the edges. Okay? So it turns out that if you work out one standard deviation, okay, just sigma, not sigma squared this time, okay, was that 68% of the data will be in between plus 1 standard deviation and minus 1 standard deviation. Okay? So if you want to know how likely it is for you to, say, make another measurement of a person or of a population or anything, and it to be within a certain range, then you could use the mean plus or minus 1 standard deviation. Okay? And that would tell you that you were 68% likely to sample that again. Okay? If you do 2 standard deviations, okay, it ends up being approximately 95%. Okay, and that's quite convenient. So if you work out the standard deviation, times it by 2, add it onto each side of your mean, though that range of values, 95% of the time, if you make another measurement, it will be in that range. So it gives you some predictive capacity. Okay, so basically what you're looking at is if you measure 2 things, okay, and you plot those 2 standard deviation error bars, if they're different from each other, then it's like 95% likely that those numbers are definitely different from each other, which is kind of what we kind of want to know in science. It's kind of not quite that straightforward, but Massimo will go over more of the details of how you end up with a prediction in terms of percentage likelihood of being within a certain range. Okay, so I guess just to point out that relationship between the likelihood in terms of percent and the standard deviation only really applies if you've got this case of a normal distribution. And that's this case where you've got a mean and all of your measurements are evenly distributed each side of the mean. Okay, so you're not more likely to be skewed over to one side than the other. And you're more likely to be near the middle than the edges of the distribution. So it's quite common if you kind of like are measuring a thing, okay, you'll end up with a population that does look something like that. So if you just end up, if you basically start out with kind of, this is the number of measurements you might make, okay, and this is the value, Okay, you might start measuring, one measurement might be here, and then you might make another measurement, that might be here. Okay, but then you're going to start measuring ones that are on top of each other. The more measurements you get. So you're building up, you're more likely to make a measurement near the middle than you are at the edges. So you'll end up with if you measure infinite number of samples, you'll end up with a kind of population distribution that looks something like this. Okay? This is really common in kind of quite a lot of measurements that we make. Sometimes you might get different distributions. So you might get a distribution that looks something like this, where actually the distribution looks, so you basically get randomly numbers in between two values, okay, but there's no increased probability of being near the mean. Okay? If you've got a population distribution that looks something like this, the mean, which would be somewhere around here, does mean something. That means that that's the most kind of likely value, but it's actually not correct to do a standard deviation for this. Okay, because that doesn't tell you anything about the likelihood of being here or being here. So this is another reason why it's always important to plot up your data to see how it looks. Okay? Does it look like it's uniformly distributed, or does it look like it's normally distributed? So normally distributed is kind of like how I just showed that bell-shaped curve. Okay, we're coming towards the end, which is kind of nice. So these things like mean, standard deviation, there are functions for these in Excel. Okay, so quite annoyingly, the function for mean in Excel is called average. So if you type equals average, then draw a little bracket and then highlight the numbers that you want to average, close bracket, that will give you the mean value for those cells. There are also functions for median, which is another way of the type of average, which basically median is you rank all of the numbers from smallest to biggest and basically pick the middle value. Okay, so that's useful if your data is not normally distributed. If it's kind of skewed over to one side, it's kind of quite useful. Now, Excel used to have a function stdev to work out the standard deviation. Okay, it still does have that function, but it's now basically got two additional functions to be more explicit which standard deviation you're working out. So there's stdev, okay, which if you're measuring the whole population of something, okay, then stdev will give you the population standard deviation of everything because you're giving it everything. But if you only have a sample, okay, of the whole population, you should use stdev.s, and that uses that unbiased formula. Okay, so you have a play around when you do this in the tutorial. Try it with stdev.p and stdev.s, okay, and use the right one. They should give you different answers, okay, this one should give you a smaller answer, because this one is systematically making the answer bigger for small values of n. Okay, but you can't cheat and you make this one to give you a smaller error bar unless you're actually measuring everything in the population. Okay, if you're only making a subsample of all possible measurements or all possible things, then you should be using stdev.s. Okay. And then there's also this function normdist, which allows you to generate data in that normal distribution. So that's how I made all of that kind of fake data in that big cloud of data before. Okay, so just briefly, because I think Massimo's going to go and do a lot more of this with you. I didn't realise I had a little picture of chalk on the slide. That's odd. So this thing called correlation. So sometimes, if you're measuring two variables, okay, so if you're measuring height and weight of undergraduates, I guess, you quite often find that they're not completely unrelated to each other. So quite often, height will vary with weight. Tall people tend to be heavier. Okay? So there's this kind of thing about correlation between variables. Okay? So if there was no variation between your variables, if there was no, I guess the other thing you could measure IQ and shoe size or something like that, you might have some data that looks like this. So if I drew a graph on here, this was IQ and this was shoe size. Okay, you might expect the data just to be completely random. Whereas if you go over here and I did weight and height, yeah, I might find a tall person, they might be quite heavy. Small person, short person, they'll be quite light. Yeah. Now you might not expect them to form perfectly on a straight line. You might actually expect them to be a little bit more scattered, but still slightly related to each other. Okay? You might get some like really short, fat people and some really tall, skinny people. So we have this thing called correlation coefficient, which allows us to make some estimate of how well one variable changes with another one. Okay? So if we have a correlation coefficient of 1, then they're perfectly related to each other. If we have a correlation coefficient of 0, there's no relation between them. And if we have negative values of the correlation coefficient, that means that when one goes up, the other goes down. Okay? So when you're looking at data, you do need to be well, the best way of assessing it, first of all, is to plot up your data and see what it looks like. Okay? So there is this formula which you can use if you want to. There is a function in Excel called correlation, which allows you to work out basically the R value. Now, I'm not going to bother going through the details of that, but effectively you're looking at the difference between the mean value in X and the mean value in Y, kind of compared to the mean difference in X and Y. Okay, so you could use this function in Excel for Corel. So this is very similar to if you make a graph in Excel. You know, if you click on trend line, have you guys done that before? You will do, I promise. You get an option of showing the correlation coefficient, okay? And that's often referred to as an R squared. So an R squared ignores the fact that sometimes you can have negative correlations. Okay, it's useful for other things which Massimo will go into, but basically, yeah, so this is R squared. So it basically gives you a measure of how much one variable can explain another variable. So if you had a correlation between people's weight and people's height, okay, and you measured a sample of people and you worked out the correlation coefficient and you squared it and you got, you know, a correlation coefficient of 0.8, that says that if you wanted to go and then just measure people's height and work out what weight they were, okay, without having to measure their weights, just use the relationship from your previous estimate, 80% of the time you'll be right. That's all it's kind of like. Okay, so there's some caution things with correlations. So you can get data that does covary, okay, but it might not mean anything in terms of the real world. Okay, so the classic kind of thing is if you map out in a city where firefighters are, okay, and where fires are, okay, you quite often find them in the same place. Okay, so from that you cannot conclude though that firefighters cause fires. Okay, so there are kinds of all kind of yeah. So smoking leads to lower exam marks. Okay, so if you smoke, then that inhibits your brain function. You get lower exam marks. Does it work the other way around though? Okay, so maybe the causation is the other way round. So maybe because you've got lower exam marks, you tend to be a person that's made poor life choices, and you're then more likely to smoke. Okay, so sometimes the causation in a correlation is backwards. Okay, so you need to be aware of that when you're interpreting data. sometimes you could, have a, So school kids, shoe size is correlated with reading skills. Okay, so if you go out to a school, primary school, I don't go bothering primary school kids, but if you go and measure their shoe size and how good they are at reading, you get quite a good positive correlation with shoe size and reading skills. Now that's not because...
Are you making this up?
No, it's true. It's because older kids have got bigger feet. Yeah, so there's a third reason here, age. Yeah? So you've basically got some data, it correlates really well, but there's basically a third data set, age of child, which there is a correlation with that you're missing out on. Would I make it up? Yeah. And sometimes you can get correlations just randomly. If you measure enough things, okay, then by chance, things will correlate with other things. So basically, global warming has been related to piracy. Okay, so if you look at the population of pirates, it goes down, global warming goes up. I mean, that's not true because the population of pirates has actually gone up. This is basically the population of pirates that speak like pirates has gone down, but the population of pirates that kind of speak like Somali kind of like farmers, that's gone up. Okay, so that's just that is a made up thing because it's been on Wikipedia. Okay, so we're going to stop there because that's quite good. I think it's a good place to stop. Let's stop there. Okay, so there are some other examples there which this presentation is on the Learn, so you can go and have a look at those. I think we've done. So you guys have a tutorial later on this week, and then it's hand in a clock for your doobie Watsons. Yeah.
What?
Tutorial work. You've got to hand it in.
On Friday?
Yeah.
Yeah.
After the tutorials that you can have. Do you want to? Or you?
I think some of it will.
Okay.
Have we got another lecture, is it?
No, it's only tutorials. But you got right to do that. We might have to have this chat elsewhere.